Our next speaker prior to our break is Dr. Brenda Vincenzi. She's the Global Development Leader and Senior Medical Director at Roche Pharmaceuticals, and she's going to be speaking to us about the Roche Angelman Syndrome Program. Brenda? She has the best accent of speakers today. We all wish that we had an Italian accent.
They cannot hide that.
You cannot hide it. No, you cannot. This is
Good morning, everyone. It's a real pleasure to be here, finally in person at FAST. So thank you very much to the organizers. Just a brief disclaimer, so the following content is being provided for informational purposes only, as scientific exchange, and not for promotional purposes. Alison already introduced myself, so I'm going to skip this slide. So today we have a very rich agenda. So I'm going to give you an overview of the Angelman Syndrome Program at Roche, and then I'll talk about the ruconirsen, our antisense oligonucleotide LNA, in the context of drug development and all the steps that we have to follow. And then I'll give an update on Tangelo, our phase 1 drug study with ruconirsen, and an update on the PET study, which is an imaging study that I introduced to the community last year. And I am very pleased and excited to share with the community that Roche had decided to enrich its pipeline and to add Alagabat, which is an additional drug for Angelman syndrome deletion genotype. And I'll explain everything later. So, you know, we've been working with the community for a number of years now, almost 8 years. We developed a disease concept model in collaboration with ABOM. We started an observational study called Phrygias in partnership with Biogen and Ionis. Phrygias was completed last year. We are very close to submit a publication. And then in 2020, we started Tangelo, our phase 1 drug study with ruconirsen. And I'm pleased to say that the first part of Tangelo was completed, and we started the second part, and I'll update you on this later on. And then the PET study. So we started the PET study last year. The PET study is an imaging study, and we completed the PET with all the patients enrolled, the healthy volunteers enrolled this year. And I'll give an update later on the PET as well. And then you see in 2023, we continue the long-term extension of Tangelo, but we also start a study on Alagabat that is planning to start the first 6 months of 2023. So here you see an overview of the drug development steps that we have to take when we want, from the point when we develop a drug in the lab to the start point where when we discuss with the health authorities, we get approval and the drug can reach the community. So the first step is called discovery. And this is exactly what we did with ruconirsen. So we took cells from Angelman syndrome individuals and we developed into neurons. As expected, those neurons did not express UB3A protein. We tested a number, as you can see here, a number of LNAs. LNAs are antisense oligonucleotides that are chemically modified with the LNA lock nucleic acid chemistry to make them better. And then we identified one particular LNA, and this is called as a name, and now it's called ruconirsen. So ruconirsen is an antisense oligonucleotide chemically modified with this LNA lock nucleic acid, and this is called RNA therapy. So ruconirsen, what we call the target profile. So at the end, what do we want this drug to do and to be? So what, in this case, we talk about a label. So we aim for a broad label for ruconirsen, meaning that this drug will address Angelman syndrome as a whole. This means all ages and all genotypes with infrequent interventions. This is despite the fact you see there, Tangelo. This is the study that we are running right now, age 1 to 12, of mutation and deletion genotypes. But this is only the first step, and then we'll have additional study to achieve that broad label. So here you see the second step in orange. So after we have discovered, we have our target, we have discovered our drug, rugonersen, and then we have to test that drug in what we call the preclinical setting. So preclinical is everything that happens before the drug reaches the clinic, before the drug can be tested in humans. So we also run toxicology studies in at least 2 species, as you can see here. And one of the studies that we performed is this one that you see here. So in this study, so the summary is that rugonersen increases the UBE3A protein in cells, and this is on the right part of the slide, and in monkeys. So for now, just focus on the orange box, on the monkeys. See if I get this. Okay, so it looks like a complex study. In reality, it's very simple what we did. So we got monkeys. These are non-disease monkeys, so they don't have Angelman syndrome, and that's why if you look here, they start with a 100% expression of the UBE3A protein. So what we did, we administered 1 lumbar puncture of rugonersen, exactly what we do in the clinic. And the first thing we want to see is the antisense, so the light blue color line. We want to see the antisense, so that break that prevents the paternal allele to express the UBE3A protein. We want to see that going down, and that's exactly what we saw. But this is not enough. What we wanted to see is actually the UBE3A protein, so the dark blue line going up. And you can see like this beautiful, beautiful line and the protein that starts to going up. Now, per protocol, we stopped the study at day 85, but you can see like that this was a long-lasting effect. Right, so once we have collected all the information about rugonersen, we perform all the preclinical studies, all the toxicology studies. We put all of that data together in a big document, and we send it to the health authorities, so to the FDA and European health authorities. And we ask them, so this is all the evidence that we generated with rugonersen. Our intention is to study rugonersen in Angelman syndrome individuals. Do you agree? And that's exactly where we received the green light at this point to start what we call the clinical development part of the drug development process. So clinical development is exactly when we start dosing patients with Angelman syndrome. Now, in the classic drug development process, usually you have a healthy volunteer phase, and then you go ahead with bigger trials in patients. In this case, you know better than me by now that all of the companies that study the antisense oligonucleotide, they started with a phase 1 that is in pediatric patients with Angelman syndrome. So this you can see here is where we are with Tangelo. As Becky mentioned before, drug development is a long process. It can take up to 10-15 years to develop a drug, meaning that from the time when you discover you have your target, your molecule, to the time when you see the star, when you can have approval from the health authorities and bring the drug to the community. In rare diseases, these timelines can be shortened, and one thing that we can do usually is to join phase 1 and phase 2. So from a study like Tangelo, a phase 1 study, then we plan to go with a phase 3 study. So Tangelo, I think you're very familiar with Tangelo by now, but just a reminder, Tangelo is an open label study, meaning that everyone receives the drug. It's a multi-center study. We have centers, we have different countries and different centers around the world. We want to investigate, the first objective is to investigate that the drug is safe and tolerated, and then the PK. So PK means that we want to understand how the body reacts to the drug. and the pharmacodynamics, so PD, that we want to see how the drug reacts toward the body, clearly with rugonersen in participants with Angelman syndrome. We enrolled ages from 1 to 2. The genotypes included are mutations and deletions, and we have 4 countries and 10 sites active. So the study duration, we started in 2020. I put here an approximate date for 2023 at the end of the study, and this will be based on data coming from the long-term extension, so the second part of the study that is currently ongoing. I'm glad to share that we look at the data, and rugonersen has an acceptable safety profile. This, in conjunction with positive preliminary data coming from the study, led the team to decide to plan the next phase in the clinical development of rugonersen. So this is the first part of the study that is now completed. The first part is also called multiple ascending dose, and this informed us on the highest safe doses that can be administered to patients with Angelman syndrome. So the higher we go with the dose, the longer is the interval between lumbar punctures. And now I explain exactly how we did it. So we started with very low doses. So we started with the 5 to 12. So we enrolled a small number of individuals in what we call a cohort. A cohort is simply like a small number of individuals with Angelman syndrome, age 5 to 12. So we started by administering very low doses, and we escalated these doses. And you see, so we started with the first dose in this patient. Then after 4 weeks, we administered a slightly higher dose to the same patients. And then after 4 more weeks, we administered a third dose that was slightly higher than the previous one. So once we have completed this cohort, we look at the data. Everything looked okay, looked good. So we decided to progress with the next cohorts. So we unlocked another cohort in the 5 to 12, but we also started with these very small children, age 1 to 4. And we did exactly the same thing. We started with these are new patients, new participants. So we started with 1 dose, 4 weeks, a second dose, slightly higher than the previous one, and a third dose. At the end, we look at the totality of the data. Up to this point, everything looked good, so we decided to progress with 2 additional cohorts. We did exactly the same thing until we completed the MID part of the study, or first part of Tangelo. And this is fully enrolled with 50 participants enrolled in the MID. Then what we did is that we gave to some participants, in particular the ones that were enrolled in the first cohorts, we gave what we call a bridging dose. A bridging dose is an additional dose. Look at this as a booster dose to bridge between the part 1 and the second part of the Tangelo that is called long-term extension. So the long-term extension is currently ongoing. We are administering different doses with longer intervals that we have tested previously in the MID part of the study. So right now we are testing these doses every 16 weeks, which is approximately 4 months, and every 24 weeks, which is approximately every 6 months. And we want to look at the safety and tolerability with a chronic exposure to rugonersen. So, and now it's like this big question, like this big elephant in the room, elephant that I hope that is becoming smaller and smaller every time that I talk about why Roche is not sharing data. So I appreciate the Ultragenyx point of view. I have to say that I share a different point of view. There are clear scientific rationale for not sharing data. And so the question is, why Roche is not sharing data while the trial is ongoing? And the answer is very simple. because the trial is ongoing. You do not share data while the trial is ongoing. This is because you can get if I share numbers and data now with you, even if you don't want to, there are expectation biases that can really influence the outcome of the study. And we are using the outcome of the study to plan the next study. So I don't want to put the outcome of the study at any risk. So in placebo-controlled studies, the drug has to be better than placebo to be successful. We have an example. This is the gaboxidol trial that unfortunately failed not that long ago. So if you're familiar with the trial, and when they presented the press release and when they presented the data, they presented a robust placebo effect. What does that mean? That means that the individuals that were treated with gaboxidol, individuals with Angelman syndrome, and individuals that received the placebo, so the sugar pill, not the active drug, they both got better. And you might think, well, but that's fantastic, everyone got better in this study, but that's exactly the point. Like, this trial failed. This drug did not reach the community, the Angelman syndrome community. So even if Tangelo has no placebo, it's an open label, like the other phase 1, phase 2, 1, 2, they are ongoing. Still, there are positive or negative beliefs about the drug that can influence or skew the outcome of the study. So I'm asking the community to stay objective as much as we can. We all have to stay objective. We all want to bring a drug to the community, but we do have to stay objective. Roche is not sharing data for, like, the fact that we are not sharing data doesn't you shouldn't reach any conclusion. It's not like a good sign or a bad sign. We are committed to transparency. You know, there is no paternal view toward the patient because we are not sharing data. That's very far from the reason why we are not sharing data while the study is ongoing. And we will share data as soon as possible. So this brings me to another very important point. That is the role of the caregivers once you decided to enroll in a clinical trial. So information about the trial, experience during the trial, should not be shared with other families. I totally understand the need for families to connect with other families, and this is totally okay. What I'm asking families that are enrolled in these trials is not to share data with other families that are enrolled in the same trial, or with the community. Don't post on social media what you think that you have observed efficacy-wise or safety-wise in your child. This clearly can influence, as I said before, the outcome of the trial. So when you are about to share something, ask yourself, what is the benefit of sharing this information with the community? Is there any benefit if you look at the big picture? And the answer is 100% no. Right, so now we switch topic and we go to the PET study. So the PET is an imaging study that I introduced to the community last year. And we completed the study. I can tell you a little more about what this study is and why we are doing it. The PET is an imaging study in healthy adults. So these are individuals, are adults without diseases. And we administered, I can show you, okay, here. It looks complex, but I guide you through this. So a PET study is an imaging study. You're probably more familiar with the MRI studies, with TC scan, but that's exactly the same family. These imaging studies are used to see what's going on inside our body. And this is what we did. So we enrolled healthy adults, healthy volunteers, and we administered through a lumbar puncture rugonersen, a very low dose of rugonersen, so the same drug that we are using in the clinic, in our Tangelo study. But this drug is a radio-labeled rugonersen. imagine like that this drug, the rugonersen, is wearing like a fluorescent backpack. So wherever the drug goes, you can always spot where it is. So after we administer the lumbar puncture with this radio-labeled rugonersen, we put the healthy volunteers into a PET machine. It's a big machine that takes very fancy pictures, as you can see here. So we took different pictures, and then you can see, if you think about the fluorescent backpack, you see where the drug ends up being. So you see that at the very beginning, this is just an example. At the very beginning, when you administer the drug through lumbar puncture, you see the drug in the spine. And then with time, you see that actually the color goes up. And the final one, you see this beautiful picture where you see the drug in the brain. And that's exactly where we want the rugonersen, so the drug, to be. So where are we the study? We completed the study. We enrolled 20 healthy volunteers. We performed 152 full body PET scan. We are analyzing the data. And of course, we have a lot of data that we are analyzing now. And the data will be shared with the community in due time. So why do we perform such a study? So this is the first study performed with an antisense oligonucleotide administered intrathecally in humans. So we don't have, up to this point, we don't have information coming from human beings. The only thing we had up to this point was a similar data, but coming from animals. And that's exactly why we are performing this study. And that's exactly why we are looking at this study as a real pioneer work that will inform other LNA or antisense oligonucleotides drugs that are administered via lumbar puncture. So what we want to understand, once we administer the drug through lumbar puncture, where does the drug go? And that's one of the objectives. One other objective is that we test a different procedure to see if we can see a different biodistribution of the drug in the body, so in the central nervous system of healthy volunteers. All the learnings coming from this study is now helping Tangelo and will help the next phase or the next study in the clinical development of rugonersen. So and now this brings me to Alagabat that I introduced at the beginning of the talk. So Alagabat is a new investigational drug for Angelman syndrome individuals with deletion genotype. So before I talk about Alagabat, I want to address the community, because clearly some families here, they probably focus on one word, and this word is deletion. And the question is, what about the other genotypes? Why there is another study on deletion genotypes? So for Alagabat, there is a clear scientific rationale for studying this drug in deletion. But I want the community to remember what I mentioned about rugonersen. So the aim for rugonersen is to have a broad label, to have a drug for Angelman syndrome as a whole, including the other genotypes and all ages. So Roche has not forgotten about the other genotypes. So we know, and this community is so well educated, and we know, you know better than me, that individuals with deletion genotype, they present generally, they're more impaired than the other genotypes. There are deleted genes beyond the UB3A gene that might contribute to the pathophysiology, so the more severe phenotype that we observed in deletion. And here I put like a couple of examples. So if you look at the Bailey here, this is one of the scales that we commonly use, right? And I put here the cognition domain, but this is also true for the other domains, so communication and motor skills. If you look at the red lines here, the red line represents the deletion genotype, and then you have in blue the non-deletion, so the other genotypes, and in gray here the normotypical, so children of the same age without Angelman syndrome. And what you can appreciate that in general, individuals with deletion genotype, they present with scores that are lower than the other genotypes. The same is true for epilepsy. So you observe here in red that the onset of seizures and then receive a diagnosis of epilepsy later on is earlier than the other genotypes. You see this steep curve. In general, they present with more seizures, and the majority of deletion genotype present with they have a diagnosis of epilepsy. So overall, there is a more severe clinical picture. So a deletion, when we talk about deletion, what's a deletion? Deletion means that there is a large portion of the genetic material that is gone, is erased. And this includes not only the UB3 gene, but also other genes. And part of these genes are the UB3, the GABA receptor genes. So here you see part of the chromosome 15 in a normal, typical, so an individual without Angelman syndrome. So here you see the UB3A, so the maternal and the paternal. So the maternal that expresses the UB3A, as we know, and the paternal that is silenced. And then you have all these other genes. If you look at mutations, so individuals with a mutation genotype, you see that the maternal allele of the UB3A is mutated, so it cannot express protein or functional UB3A protein. And then you see the paternal, as we know, is silent, so the paternal cannot express the UB3A protein. And then you have the deletion genotype. Regardless of the size, we have different size of deletion, but regardless of the size, you see here that the UB3A gene, here the maternal one, is deleted, but there are other genes that are deleted as well. And some of these genes are the GABA receptor genes. GABA receptor genes, they are extremely important for brain development and neuronal function. So if you look at here, these three blocks, and now you look at this slide. So these three blocks, these three genes, maternal genes, they are deleted on the maternal allele of deletion, individuals with deletion. So these genes, they have the recipe inside them to basically produce this protein, this colorful protein. This protein, they get together in a sort of a little puzzle, and they make this GABA alpha 5 receptor. This is one of the subtype of the GABA receptors. So think about the brain, right? You have the brain, you have neurons, and the neurons, they communicate to each other using neurotransmitters. One of the most important neurotransmitter is the GABA that you see here, this yellow dot. In order to communicate, this GABA has to bind to what we call a receptor, which is as simple as a protein. And this receptor is basically located on the surface of the neurons. In Angelman syndrome deletion genotype, because of the deletion, there is a reduced number of GABA alpha 5 receptor subtype. And this might be linked to the more severe phenotype that we observe in these individuals. So the GABA R receptors are important for brain function, as I mentioned, and brain development. In Angelman syndrome deletion genotype, mainly the GABA alpha 5 receptors are affected. Any dysfunction around this receptor has been linked with many neurodevelopmental disorders and with epilepsy. So our hypothesis for deletion Angelman syndrome genotype, while correcting the UB3A loss is key for treatment of Angelman syndrome, including the deletion genotype. A therapy that selectively enhances the GABA alpha 5 receptors functioning might improve some of the symptoms and potentially aid brain development in Angelman syndrome individual with a deletion genotype. So how do we do that? That's Alagabat. So Alagabat is an oral tablet that is taken once a day, and this drug enhances the GABA alpha 5 receptor function. So Alagabat has been investigated already in healthy volunteers, so we have data in healthy volunteers, and is also under investigation in a different trial for autism spectrum disorders in adolescents and adults. So because we have this data, we can already say that Alagabat has shown an acceptable safety and tolerability profile to date. And here you see Alagabat in the context of the receptor and the GABA neurotransmitter. So you see exactly where the Alagabat binds, and once it binds in the presence of the GABA, so the neurotransmitter, it helps the function of the receptor itself. So Aldebaran is the name of the clinical trial that we are going to start with Alagabat. That is the name of the drug. It's a phase 2A study in individuals with Angelman syndrome deletion genotype. It's an open label study, so there is no placebo. 12-week study to investigate the pharmacokinetics. So we want to understand, once again, what the body does to the drug once the drug is administered. The safety and tolerability, this is always a must, and what we call a proof of mechanism of Alagabat. So we are going to test, that means that we're going to test different doses of Alagabat, and we want to look at the signal at the EEG. So we are going to enroll up to 56 individuals with deletion genotype, both females and males. Genotypes is clearly deletion. And location will have up to 6 countries and up to 20 sites. Study start is the first half of next year. And I actually like to thank the patient council. So at Roche, we set up a patient council with the representatives, so caregivers to Angelman syndrome individuals that represent different countries, US and non-US, and we consult with them. We started more than 2 years ago. We consult with them on a regular basis. They were deeply involved in, and they reviewed the protocol for the Alagabat trial. And I'd like to now introduce the Alagabat team. So some of the team members overlap with the rugonursing, so the team that I'm leading. So all the others are new members. So Tom Anvise is the counterpart, is the medical director for the Alagabat trial. Lorraine Murtagh, that is present here in the room, is the project team lead. Jörg Ip is the biomarker lead, working on the rugonursing and now on the Alagabat program. Maria Clemencia Hernandez is the research project leader, and Shady Sedom, that is our global patient partnership director, working on rugonursing, Alagabat, and DUP15Q.